Good morning, children. Our topic is crossing over. In the first point, introduction, the interchange of chromosomal parts between non-sister chromatids of a homologous pair of chromosomes resulting in recombination of genes is known as crossing over. And in crossing over, the crossing over was discovered by a scientist known as Morgan. Crossing over occurs during meiosis stage only. That in the meiosis occurs in the gametogenesis. Okay. Next one, formation of the gametes is known as a gametogenesis. In that gametogenesis, meiosis occurs. In that meiosis, the crossing over takes place. Next one, the number of crossing over depends upon the length of the chromosome. These 4 points you have to write in the first point subheading known as the introduction. And let us name second point is the mechanism of crossing over. Okay, in the mechanism of crossing over is the mechanism of crossing over. So what are the points are present in the mechanism. Here crossing over occurs during meiosis of the gamete as usual. Then the crossing over occurs in the Pectin stage of prophase 1 in meiosis. Okay, what are the steps are present here? First of all, in zygote stage of prophase 1 meiosis 1, the homologous chromosomes move towards each other and come to lie side by side. This is the first step. See here, these are two different chromosomes, they're coming to lie side by side. Okay, this phenomena of pairing homologous chromosomes is known as a synapsis. The paired homologous chromosomes are called the bivalence. These two are called the bivalence. And the second step, the homologous chromosomes split longitudinally. So here this one chromosome splits into two. This another chromosome splits into two, like this. Here split into 2 chromatids, hence 4 chromatids are produced from the 2 homologous chromosomes. This stage is known as a tetrad stage. The 2 chromatids of a chromosome are attached by a single centromere. See, these are the 2 chromatids of the single homochromosome. These 2 chromatids are attached to by a centromere, like this. Okay, now the 2 chromatids of chromosome are attached by a single centromere. Now, the non-sister chromatids of homologous chromosomes twist over each other. Okay, this is the next step. What happens here is one chromosome is overlap on the another chromosome. Okay, now at certain points the non-sister chromatids are connected with each other, so that when one chromosome is overlapped on another chromosome, here they connect with one another in the form of the X shape. The points where the connect taking place between the non-sister chromatids are called the chiasmata. Okay, chiasmata means a cross. And next one, what happens after the formation of the chiasmata? At the chiasmata, the chromatids break. This is brought about by an enzyme known as the endonuclease. Here, when the attaches with one another, each chromosome breaks like this. Here, one chromosome break and this chromosome also breaks. Okay, with the enzyme known as the endonuclease. The broken segment of the chromatid is fused with the other chromatid and vice versa. This fusion is brought about by another enzyme known as the ligase. Hence, the process is known as the ligation. Okay, thus the exchange of the chromosomal segments between non-sister chromatids is known as a crossing over. Then, last step after crossing over, after the exchange of the chromosomes, genes, here what happens after crossing over? The non-sister chromatids repel each other. That means they separate with one another. They separate from each other from twisting. The separation of the starts from the centromere towards the ends like a zip. This separation is known as the terminization. Okay, next one, third side heading is crossing over mechanism theories. Okay, there are 3 theories are present. We will discuss very briefly. First one is a chiasmata type theory. The theory was proposed by Jensen. This theory suggests that prior to crossing over, the chromosomes of each bivalent get to duplicate to form a tetrad, as I said in previous point. After formation of the tetrads, the crossing over occurs only between the non-sister chromatids of a tetrad. In the diplotin stage, the non-sister chromatids overlap with one another and form chiasmata. In the chiasmata, what happens? The chromatids break and the region with the mutual exchange of the segments. The other chromatids remain intact. So, That means in the chase matter formation, the exchange of the genes takes place. That theory is known as the chase matter type theory. And the second one is the duplicate theory. This theory was proposed by John Belling. Belling believed that crossing over might occur during the duplication of the chromosomes. That means when the chromosomes are divided into two, like this, what happens when they divide into two, then there only the exchange of the genes takes place. So but he brought about due to the novel attachment found between the newly synthesized genes. Next one, last theory was break and exchange theory. The break and exchange theory is the most accepted theory to explain the process of the crossing over, not only with the chase matter formation, not only when the two chromosomes are duplicated, when the break, then only the genes exchange takes place. So that's why this theory is more accepted. So this theory states that in the crossing over, breaks occur in the non-sister chromatids of the tetrad. Then only the exchange of chromosomal segments occur between the non-sister chromatids. These three theories are presented to explain the mechanism of the crossing over. Okay, next one, fourth one is the kinds of the crossing over. There are three types are there. Number one, single crossing over. Number two, double crossover. Number three, multiple crossover. Okay, let us discuss how many types are there. Here, according to the number of chasma, according to the number of how many chasmas are formed, according to that, the crossing over is classified into three types. Okay, single crossing over. If the chase matter occur only at one point of the chromosome pair, then only the crossing over is known as a single crossing over. And next one, if chase matter occur at 2 points of the same chromosome, then that phenomenon is known as a double crossing over. And then multiple crossing over, when crossing over takes place more than 2 places in the same chromosome. If chase matter are formed more than 2 places, then the crossing over takes place in that 3 points. Such type of crossing over is known as a multiple crossing over. And what are the factors affects the crossing over? Here's the first 7 points are there. First one is X-ray increases the frequency of the crossing over. Okay, and next one, the high temperature also increases the frequency of the crossing over. Next one, the frequency of the crossing over, what happens, it decreases the increasing age. And next one, crossing over is less frequent near centromere. That means in the centromere, the crossing over does not take place. Only at the tips of the chromosome, the crossing over takes place. Some genetic mutations also decreases the frequency occurs of the crossing over. Next one, inversion of chromosome segments suppresses the crossing over. So when an inversion takes place in the chromosome, so this suppresses the controls or decreases the crossing over. And chase matter formation at one point prevents the chase matter formation in the vicinity. So this phenomenon is known as the interference. Okay. Next one, what is the significance of the crossing over? Here 4 points are there. Crossing over clearly illustrate that the linear arrangement of genes in the chromosome. That means in the chromosomes, what happens is the genes are arranged in the linear way. Next one, the frequency of the crossing over is very useful to construct the chromosomal maps. So that according to the chromosomal maps, we can we can identify the structure of the chromosomes, the number of the chromosomes, the disease of the chromosomes. Okay. Next one, crossing over produces a new combination of the genes. Okay, crossing over leads to genetic variation. These are the significance of the crossing over. Okay, thank you.